Biomarker has pioneered the development of genetic technologies and industry since 2009 and owns more than 60 invention patents and over 200 software copyrights. Additionally, Biomarker has contributed to over 1,000 scientific programs published in high-level academic journals with a cumulative impact factor of more than 4,500. This continuous innovation in scientific research fuels Biomarker to become one of the premier promoters of genetic technologies in the era of biotechnology. Biomarker aspires to simplify gene decoding and accelerate advances in scientific research and is committed to provide standardized, customized, and comprehensive technical services and solutions to customers all over the world. We have established 4 business segments devoted to high-throughput sequencing, bioinformatics, biocloud, mass spectrometry, and intelligent manufacturing. In the field of next-generation sequencing, Biomarker leads functional genomics with SLAF-seq. SLAF-seq has accurately identified hundreds of vital functional genes that closely relate to human lives and has served a role in over 30,000 projects. Since third-generation sequencing came onto the stage, Biomarker became one of the earliest players in this new arena. We are proud to have introduced the PacBio platform-based tandem full-length 16S, 18S, ITS, amplicon sequencing technology and have launched Nanopore platform-based full-length transcriptome sequencing services, which brings microbiome and transcriptome study to a new level. The internet era has provided great opportunities in genetic technologies. Biomarker has efficiently and professionally integrated multi-omics-based big data and an online interactive analysis platform and high-performance cloud computers on BMK Cloud, a self-developed online biocloud system. The platform is designed to help users interpret biodata and promote scientific research and health management, leading the generation of cloud-based genomics and biodata, guided by the core values of continuous innovation and the pursuit of excellence. Biomarker provides its customers with high-quality services and products. At the same time, Biomarker has built state-of-the-art and automatic professional molecular experiment platforms, automatic and intelligent bioinformatic analysis platforms, and an information-based and standardized system to manage production, sales, staff, and finance. Biomarker will continue to create greater value for its customers and lead the industrial transformation with high-quality innovations to realize its dream of benefiting humanity with genetic technology. Biomarker Technologies. Innovation leads to wonders.
Hello, everyone. Welcome to join today's webinar. My name is Tina Ding. I serve as Regional Sales Director at Biomarker Technologies in America. In this region, our services covers United States, Canada, Mexico, Argentina, and Colombia. In today's webinar, we have 2 sections. The first one, I will introduce our company, Biomarker Technologies. In the second section, our field application specialist, Dr. Kevin Bradmere, will present the introduction of our self-developed BMKMANU S3000 chip and spatial transcriptome sequencing technology. Biomarker Technologies, we also call it BMKGENE. Before starting to know much about BMKGENE, I would like to start with a couple exciting and encouraging numbers. The first one, 15 years. We have more than 15 years history in this field. We are a pioneered player in the biotechnology industry, and BMKGENE possesses over 60 invention patents, and over 200 software copyrights. BMKGENE has more than 500 employees globally. In addition, BMKGENE has contributed to over 1,000 scientific programs published in high-level academic journals. The total impact factors is more than 4,500. Next slide, Kevin. BMKGENE is global presence, and our clients from all over the world, over 50 countries, and we have 4 laboratories globally. As I mentioned, we have 15 years history. It was founded in Beijing, China in 2009 and has more than 15 years history, although it's new in United States. Next one, please. So this is our high throughput sequencing platforms, and I would like to share with you the platforms and our capabilities. For the short read sequencing, we have Illumina NovaSeq X and Illumina NovaSeq 6000. For the long read sequencing, we offered PacBiome Review and PacBiome Sequel 2. For the single cell support, we have several options. Like today, Kevin will give the introduction of the BMKMANU S3000 spatial transcriptome sequencing. Next one, please. We can clearly see that BMKGENE almost offers all kinds of the sequencing services, like the transcriptomics and epigenetic sequencing, plant and animal genomics, microbial genomics, human genetics. And also we have our self-development bioinformatic analysis platform, BMK Cloud. I will introduce it later. Please, next one, Kevin. This whole workflow introduces our services from the experiment design to after-sales support. Our clients, based on their sequencing needs and their research objective and goals, they can start from any step. And we can start with you from the experiment design, and also you can directly send us the tissue samples. We will perform DNA and RNA isolation and take care of all the whole procedures. Of course, you can also send us the DNA and RNA samples. We will focus on the library preparation, sequencing, and data analysis. Some clients, they provide the pre-made library. We offered this service to our clients. Next one, please. This is in the beginning, I mentioned the BMK Cloud, the platform. This is our self-developed, the bioinformatic analysis platform. It covers very powerful analysis, the tools, and also several the user-friendly apps. Based on your sequencing needs and all your research goals, you can customize analysis. You can make the beautiful graphs and figures and tables for your manuscript submission or the grant submission purpose. You don't need to have the computer science background. You don't need to write code. In the platform, we have very popular apps such as amplicon sequencing and like the 16S, ITS, and like the shotgun metagenomic sequencing, as well as the bulk RNA sequencing with and without reference genomes. Next one, please. So this is another interesting part, exciting, and I'm proud to share with you our business development in United States. I'm the first employee in United States, and we started in February. Since we signed the first contract with a client from Yale University on May 23rd, 2024, until today, right now, we have signed 106 contracts and successfully complete 49 projects. Our clients are from the research institutions, universities, the companies, as well as federal agencies, such as the USDA. So I will stop here today and let's move to next section, the introduction of BMK Cloud S3, BMKMANU S3000. Before transfer to Kevin, I would like to introduce Kevin's background. Dr. Kevin Bradmeyer earned his PhD degree in genetics from Texas A&M University in 2021. Kevin possesses over years expertise and experiences in genetics, bioinformatics analysis. Before joining BMKGENE, Kevin had worked 3 years as a bioinformatics scientist in catalytic data science company. At BMKGENE, Kevin serves our field application specialist. He provides technical support to our clients before and after sales. Now, Kevin, please take it over.
Sure. Thank you, Tina, for that introduction. And thanks everybody for coming today to listen to more or learn more about the BMKMANU S3000 chip and how we're using it to achieve subcellular resolution. So here's a webinar outline of what we're going to go over today. So first, I'll give a quick introduction on spatial transcriptomics. Next, I'll give you some of the details of the S3000 slide. And then I'll go more in depth on some of the S3000 features, some of the more complicated features. And then following that, I will describe the S3000 workflow and what you can expect whenever you decide to work with BMKGENE. And then I'll conclude with some demo results. So first, a quick introduction. So I wanted to just quickly introduce spatial transcriptomics and sort of how it fits in with the older transcriptomics technologies. So the most common and probably most familiar with many of you is bulk RNA-seq. So with bulk RNA-seq, as you well know, it takes the average gene expression profile of all the cells within a tissue. And so this is fine whenever the tissue is very homogeneous. However, it can become a problem if you have a really complex tissue or organ system where there's multiple different cell types. So we're unable to see the expression profile of individual cells. And so this is where single cell RNA-seq came in. So with single cell RNA-seq, we're able to actually gauge the gene expression profile for each individual cell and then determine what gene cell types or what cell types are present, as well as quantify the amount of each cell type. However, this does not tell the whole story because we don't know where these cells were located in the original tissue. So this is where spatial transcriptomics helps fill that gap. So with spatial transcriptomics, not only can we get a gene expression profile for single cells, but also we can tell where the cells were located in the tissue structure. So I just want to quickly cover some of the fields where spatial transcriptomics is really being utilized. So in oncology, one example is in the tumor microenvironment. So in a tumor, cells are rapidly undergoing mutations and also interacting with somatic cells around them to generate a really complex transcriptomic environment. And so spatial transcriptomics is really useful for sort of mapping these interactions and where these cells are located. In neuroscience, the brain is a highly complex organ system composed of multiple cell types that are organized in a very complex way. So as you can see from the histology slide in A, there's many different layers on top of each other, with some of these layers being extremely thin, but very important. So with spatial transcriptomics, we can actually map where all these cell types belong and what other cell types they're interacting with. With developmental biology, we can actually look at not only spatial transcriptomics, but we can look at the temporal change as well, where we can see across different time points how cells are undergoing differentiation and morphogenesis and how they're contributing to the overall complexity of the final tissue structure. With immunology, we're able to use spatial transcriptomics to perform immune landscape profiling, as well as trace activity of immune cell activation. And then finally, just wanted to quickly point out that with the S3000, we're not just limited to animals. So we're actually able to capture transcripts from plants as well, where we're able to perform similar features to where we can look at cellular differentiation as well as organogenesis. So next, I'm just going to give you some S3000 slide details. So here on the far left is a depiction of an S3000 capture slide. Each of these blocks represents a capture area. And then if we'll take a look, the default capture area is 6.8 by 6.8 millimeter. And if we take a look at this capture area, we can actually see that's composed of millions of little spots. So in the S3000, there's 4.14 million spots total. And if we look closer at these spots, you can see that they're actually little beads that are 2.5 micrometers across and 3.5 micrometers distant between one another. So this gives us our standard 3.5 micrometer resolution. And if you will take a closer look at these beads, we see that they're actually covered in oligos. So each of these oligo sequences is composed of multiple different units, the most significant being the spatial barcode. So this barcode is important for helping us identify where each bead is located on the capture area. Additionally, we have a unique molecular identifier that lets us identify whether transcripts were of biological relevance or whether they were duplicates of PCR. And then finally, and most importantly for our chip, is the poly-DT tail. So unlike other similar offerings in spatial transcriptomics, we are not species specific to human or mouse, and we do not depend on a panel of pre-made transcripts. So we can capture any transcripts from any species and any transcript as long as it has a poly A tail. It does not have to be known ahead of time. So next, I just want to quickly go over what changed between the progenitor of the S3000, the S1000. So between the two, we've doubled the number of capture spots. And so this was accomplished by decreasing the distance between each spot. And so this has increased our resolution from 4.8 to 3.5 micrometer. But what does increasing resolution really benefit from? So here we can see that you gain increased precision on identifying which beads are associated with which cells. And in addition, you have more beads within each cell. So that allows us to capture more transcripts. So between the S1000 and 3000, by increasing the resolution, we actually were able to capture between 30 and 70% more transcripts per cell bin. And in turn, that allowed us to annotate or capture more than 30 to 60% genes per cell bin. So next, I'm going to cover some of the S3000 features. So I broke this out into 4 different sections. So first is a closer look at subcellular resolution and what do we mean by that and what are the benefits. The next is multi-platform compatibility. Following that, I will discuss flexible experimental design of our chip. And then finally, I'll describe the S3000 in a 3-in-1 slide and what that means. So first, I just want to quickly discuss tissue structure. So here is a standard H&E slide of a tissue, and here you can see the various cell types located throughout the slide. And so at 100 micrometer resolution, we see that there's many certain scenarios that can occur. So in an optimal scenario, a single bead is associated with a single cell. So in this case, you're getting the transcriptome profile of a single cell in one bead. However, you can also have instances where if the bead is too large, where it overlaps multiple cells. So in this case, this bead's associated expression profile will actually be more like bulk RNA-seq, and it'll sort of be a hybrid expression profile. And so this can lead to having clusters that aren't necessarily real biological cell types, but are an amalgamation of cell types. In addition, whenever you have really low resolution, you tend to have difficulty getting a large number of clusters because it's hard to differentiate between the cell types. So whenever we go down to 3.5 micrometer resolution, which is offered by the S3000, we can actually more easily see the cells across the slide, identify the different cell types, in the clusters, and as well as see any kind of smaller scale complexity going on in the tissue. So you can see we have a far greater number of clusters called as well because of the higher resolution. So next is just a real world example of why this would be significant. So in this case, we're looking at immune infiltration in lung. And so as you can see at the higher or at the lower resolution of 100 micrometer, you kind of see 3 main cell types. So the orange, which are associated with tumor cells, the green, which are associated with lung cells, and then these dispersed cells, which are a different cell type altogether. So when you look at this, you only see 3 major cell types. However, whenever you increase the resolution to 10 micrometer, you're really able to see a lot more cell types, a lot more clusters, and you're also able to see how these smaller clusters of cells are dispersed throughout the tissue and throughout the tumor. So here you see a lot of little teal clusters in addition to the normal maroon clusters we saw in the previous image. And so here you can actually see that these are mesenchymal cells as well as macrophages. It's just they're normally so dispersed that a larger resolution would not allow you to see those. So next, I want to discuss multi-platform compatibility. So in addition to short read sequencing, we also can perform full-length sequencing on the spatial transcriptomic transcripts. And so the benefit of this is whenever you use short read sequencing, it's normally incredibly difficult to identify, or impossible, to identify which reads belong to which isoform. However, whenever you perform capture full-length transcripts with nanopore, for example, we're able to see that each individual read corresponds to an entire isoform. So this allows us to perform isoform quantification, as well as alternative splicing. And then finally, we're also able to identify fusion genes. So this is just a quick overview of how the two technologies relate to one another. So as you can see here, if you're trying to decide between Illumina and nanopore, here's a correlation analysis. First, for the number of transcripts detected, as you can see, they correlate almost perfectly, so you're not losing any data specificity by going with one or the other. And the same for the number of genes detected per spot. And then also, for the dimensionality reduction analysis, UMAP and t-SNE plots, you're able to see that Illumina and nanopore, the cell types cluster very similarly. So this is just to provide confidence in the idea that you're not losing specificity by going with these longer reads. You're just gaining isoform level information. So next, I'll just cover some of the flexible experimental design aspects. So here on the left is the slide from the first image, or from one of the first slides, where we see an S3000 chip, and it contains multiple capture areas. So we can actually decide there's no lower limit or upper limit on the number of capture areas you can put on a slide. So if you only have one tissue you want to use, don't worry about having to use 6 different capture areas. This is easily modifiable. As I mentioned before, the default capture area is 6.8 by 6.8 millimeter. And then we also offer an 11 by 11 millimeter, as well as a 15 by 20 millimeter. And so this is important because different tissues and different target areas might be different sizes. And so this allows flexibility for getting the most efficient experiment possible. So we aim to have between 75 and 50% coverage of the tissue on the capture area. This is just to make the experiment as efficient as possible. You don't want to waste an entire capture area and only, be using 25% of it. So we also do suggest, if you do have really small tissues that wouldn't take up much of the capture area, maybe you can stack those or orient them in a way that we can utilize as much of the capture area as possible. So next, I want to discuss the idea of multi-level resolution. So here we have a standard H&E stained slide, and as you can see, the multiple cell types. Whenever we do spatial transcriptomics at the lowest resolution, we see that each of these individual spots is being clustered separately, but this isn't really telling us much about the cells themselves because it's such a low resolution. So in this case, we can actually generate subspots by binning multiple spots into these larger bins. So here, as the subspot level increases, you're actually getting bins with more and more of these spots associated with one another. And so by doing this, we're able to increase the resolution in silico from 5. So now if we look at the 10 micrometer resolution, we can actually start to see different cell types more clearly with the green clustering and the blue clusterings, and then so on and so forth. You can increase the resolution or decrease the resolution, to whatever you'd like, and whatever answers your experimental question the best. So finally, I want to cover what we mean when we say the S3000 is a 3-in-1 slide. So before that, I want to quickly go over the difference between clustering and single cell segmentation. So this is an important distinction between the two different analysis types going forward. So here in standard clustering, you have different cell types overlapping the capture area with these different spots. And so whenever you do standard clustering, you're just randomly assigning subspots in a grid pattern with a subspot. So in this case, this subspot contains 7 different spots. And so in this case, the cell only overlaps 4 of those. And so this might provide too little data to call this, to cluster this subspot. Ideally, it would cover the whole cell and it would be able to be confidently called. However, a bigger issue is many times as these subspots get larger, at lower resolutions, you can actually have spots cover multiple cells. And so, as I sort of mentioned in the earlier slides, this can lead to sort of a hybrid cell expression profile that doesn't represent accurately either cell type. It's sort of a combination of both. And so this sort of becomes like old-fashioned bulk RNA-seq, where each subspot is actually varying proportions of different cell type expression profiles. And so here we look at clustering across a tissue slide, and you can see that there is a general patterning of the different cell types as clusters, but it's not that clear what's going on. So with cell segmentation, we actually identify and annotate the cellular boundaries before doing clustering analysis. And so here we only associate spots and subspots that are associated with a single cell type or a single cell. And so here we can see only the red spots are counted for the red cell, yellow and green, and so forth. And so what that does is that allows us to more clearly identify cell types in the tissue. And you can see that it really helps illustrate the tissue complexity a lot better than the top image. So how do we achieve this cellular segmentation using the S3000? So first, whenever we take your tissue slice and put it on the slide, we'll first do fluorescent staining. This identifies the nuclei, and then we'll do an in silico cell segmentation process using a watershed algorithm. This is then corrected by using H&E staining to get clear cell boundaries. You can see in this image. And then finally, we actually perform the transcriptome sequencing, associated with the beads. And here we aim for 50,000 paired in 150 reads per spot. That's our target. And so what does that look like when we put it all together? So here we have a cross-section of a mouse testicle. And so as you can see, it's a highly complex tissue. As many of you might not know, it contains all the different cell types associated with spermatogenesis, which are highly differentiated transcriptomally and also highly morphologically different. And then also there's a whole bunch of cells that are associated with supporting meiosis and spermatogenesis. So as you can see, the spatial transcriptomics using cell segmentation really allows us to get an idea of where these cells are located, what they're interacting with, and also maybe how these cells are differentiating across time. So finally, I'm going to cover the S3000 service workflow. So whenever you work with us at BMKGENE, this is what you can expect from the process. So above is our workflow. So quickly, I just want to cover what we expect from samples. So we expect fresh samples embedded in OCT medium. In addition, you need to select a cassette that is corresponding to whatever capture area that you are selecting. Additionally, if you can provide us any information on orientation of the tissue, maybe a picture before the OCT medium has fully set up so we can see how the tissue is set up in the cassette. And then also any kind of additional information you have from previous publications or from the literature showing what your target area might look like and what areas you're interested in advance. We also suggest 2 blocks per sample, one for the standard experiment and then another as backup, though this isn't always clearly necessary. So the next and probably most important aspect of this workflow is the pre-experiment. So in this section, we do cryosectioning. So before we do the cryosectioning, we will set up a call or a live chat with you, the client. And so as we take sections, cuts, we cut 10 micrometers per section. And as we take cuts, we will stain on a normal slide and send you a picture of the cross section. And so as we start to approach the target area, we expect you to respond within 5 to 10 minutes and let us know, should we keep going or does this look good? And so we advise that you let us know if this is a good section within 30 to 50 micrometers of the target area so that we can continue to take slices to use in the actual experiment because these are just on normal slides. So using these discarded slides that are not for the capture area, we then perform bulk RNA QC to determine that the RNA quality is good. And we also perform a permeabilization optimization where we determine how long we need to let this digest to get the optimal amount of mRNA released from the cells. So following this pre-experiment, we then take an additional slice or two to use in the standard RNA experiment. Here we put the slice on the S3000 chip capture area. We do the fluorescent and HE staining, as I mentioned previously, and then we perform the standard barcoding and library prep. Here we shoot for, we use PE 150 on the Lumina NovaSeq. And then, as I mentioned maybe before, so our data output for the standard S3000 package using a 6.8 by 6.8 millimeter capture area is 250 gigabases per sample. And so at this sequencing depth, this assumes that you're utilizing roughly 70% of the capture area. So next I'll move on to data analysis. So BMKGENE offers 3 different tools for data analysis. So the first is BST Matrix. So BST Matrix allows you to go from the FASTQ sequencing data and generate a gene expression matrix at multiple resolutions. So it utilizes some of these common softwares as well as generating, like I said, multiple resolutions. So I've already introduced the subspot idea. And so this is a chart of all the resolutions that it generates automatically for each of the gene expression matrices. And so as you can see, at the lowest resolution, it contains a single spot, all the way up to the highest or the lowest resolution of 100 micrometer, and it contains a total of 469 spots per subspot. And so I also just want to mention that the gene expression matrix generated by BST Matrix is a standard default format, so it's compatible with any downstream or open access softwares like Seurat. So next is BST Viewer. So BST Viewer allows spot clustering, so standard spot clustering. So it uses a Windows user interface, and then it also directly takes in the multi-resolution gene expression matrix from BST Matrix. So it's an easy plug and play. And this allows you to overlay your gene expression matrix onto your tissue image. So here you can perform gating of a specific region of your tissue if you'd like, or you can call the clusters for the entire thing. And then you can see here that you can actually, this is where the multi-resolution aspect comes in, because you can call at, in this case, they called it level 13. And as you can see, you get 1, 2, 3 distinct clusters. And then when you zoom in on the area and call it a much higher resolution, you can see that you get some more unique clusters, whereas this orange right here is now differentiated into red and green, and you get a lot more specificity when looking at smaller target areas in your tissue. And so this program also offers flexible visualization of clusters. You can remove clusters, you can rename clusters, you can change the color. If you have marker genes, you can use those to identify which clusters are which. So BST Viewer is a very flexible tool that we offer. So next is its big brother, the BST Cell Viewer. So this takes advantage of single cell segmentation, as I mentioned previously. So here you have your H&E staining and your fluorescent staining. And then finally, those are used to perform cell segmentation. So this can be done automatically, or if you see something that looks off, you can also generate or manually draw a cell boundary yourself. This is also similar to BST Viewer. We can have, in addition, we have the H&E fluorescent image switch, so you can modify the filtering. And then also you can zoom in on a region. And then, as I mentioned before, you can call clusters. And these clusters will now be called on a per cell basis instead of as a cluster. And so here, you, like I mentioned, you can modify color, name, and remove clusters, and also change focus areas. So here's just a quick example of the BST Cell Viewer and sort of, one of the most useful aspects of being able to zoom in and having the subcellular resolution. So here you can see a mouse brain, a highly complex organ structure. So you can see many different cell types and many different layers and, many clusters called. And so here we know that this portion of the brain is actually has a lot of fine level structure with multiple different cell types. However, at this zoomed out state and at this resolution, it's really, it looks like just a single cluster. So when you zoom in and recall clusters, we're actually able to see more cell types called than there were previously. So next, I want to cover, in addition to those free software packages, here at BMKGENE, we can also do an S3000 bioinformatic analysis for you. So here we do the BST matrix analysis for you with downstream analysis in Seurat. And so here, we'll perform spot filtering, dimensionality reduction analysis, differential expression, as well as fluorescent or functional annotation, functional enrichment, some of the standard transcriptomic analyses. And so 29 analyses total with 100 plus high quality figures. So we can do this if you really don't want to, play with the data yourself too much. And so here's an example of the demo or one of our demo results from the S3000 BI package. So here's a multi-level spot clustering where we do dimensionality reduction to see the different cell types, and then how these cell types are overlaid onto the tissue. And then we do the same thing, but with single cell segmentation, where we can see the clusters called as single cells on the tissue, and then also provide all the information and details about the number of cells, genes per cell, transcripts per cell, you name it. And then finally, you can provide us a list of marker genes if you have some that are of specific interest. And here we can show which cell types or which clusters the marker genes are associated with to identify them as a specific cell type. And then we can also generate heat maps of these marker genes and where they're being expressed in the tissue. So now to conclude, I'm going to cover some demo results. So here is a demo data from a mouse brain. As you can see, it covers a lot of the capture area and is highly complex and contains many different cell types. So for this sequencing approach, we actually generated 500 gigabases of data instead of the standard 250. And this gave us a sequencing saturation estimate of 69%. And so for standard clustering at multiple resolutions, we see that the median spot at 100 micrometers is 11,000 genes. The median genes per spot at 30 micrometers is roughly 3,700 genes. And then at 18 micrometer is 2,000 genes. And then a total at the lowest resolution is 510 genes per spot. So whenever we perform cell segmentation on this slide, we were able to detect 94,020 cells. And then each cell had a median gene of 1,999. So for an even more complicated tissue, we were able to take a cross-section of the mouse embryo, the head specifically. And so here you can see a multitude of different cell types called with all the different colors. And so here the sequencing strategy was 1 terabyte of data. And so here, once again, the sequencing saturation was roughly 69%. The multi-resolution performances for standard clustering were higher because of the increase in data. And so here we see 13,000 genes, 5,000, and 3,000 for 130 and 18 respectively. And then finally at 6 micrometer, 835 genes per spot. For cell segmentation, we were able to detect 221,651 cells. And then each of those cells had a median gene expression of 1,670. So I just want to provide a closer look and make an additional case for, once again, being able to gate and zoom in on specific target regions within your tissue. So here we zoomed in on the embryonic brain. And as I mentioned before, we recalled clusters. And so you're able to see clusters that were previously unidentifiable at the full resolution. And then again, for the embryonic eye, an even smaller region where we're able to zoom in and really get an idea of the cellular structure and the tissue structure within the embryonic eye, where we were able to call 18 distinct clusters that weren't visible from the larger zoomed out cluster call. And so finally, I'd like to conclude by just saying that, as Tina mentioned, we have here at BMKGENE, we have lots of experience, so 15 years of experience, especially in spatial transcriptomics. We have experience with over 2,000 samples, and these are from more than 100 different tissue types, so we haven't just been doing them on a single tissue. And so here are just some examples and numbers from various tissue types, like mouse, spleen, and ovary. We have some breast cancer, some skin, as well as organoids. And then we've also done various other different cell types as well, or cell types and tissue types as well. So if you're interested in working with us, nothing is too strange. You can always reach out and see what we have to say about your cell type or your tissue type, and if it's feasible for spatial transcriptomics. And so with that, I'd like to thank you for your time and take any questions.
Thank you, Kevin. And we would like to take your questions. And if you have any, please type it in Q&A, the chat box.
And you're also welcome to email any questions later as well, if you don't feel like asking any today, or if you think of something later.
Again, thank you everybody to join today's webinar. We hope today's webinar was helpful and to look forward to your next sequencing project. And any questions, just like Kevin mentioned, contact us. We are always here to support. Thank you. Bye-bye.
Thank you.